Hi, welcome to the Bioinformatics Chat. Today my guest is Antoine Limasset. Antoine and I will be talking about read correction and about his new tool, which is called Bcool. Antoine, welcome to the podcast.
Hi, nice to meet you.
Let's start with your background.
So I'm a computer scientist. I started to study theoretical computer science at the ENS Cachan and Rennes in France. So I started as a pure computer scientist, and at some point I just wanted to do practical computer science, and I learned about bioinformatics because I always liked biology and genomes and stuff like this. So I just discovered the fact that we could do the both thing, and I made some internship in bioinformatics, and I just never stopped. So I continued my study in computer science, and I made all my internship in bioinformatics, working on assembly or indexation of sequences. And then I just made my PhD thesis on new methods to exploit the sequencing data using the Bruijn graphs. And I think we will be talking about this. And I just finished a postdoc in Brussels this year, working on the assembly of heterozygous genomes, so with Jean-François Flo. And I just got a position here in the French CNRS in Lille, right, starting this autumn. So I'm really, really happy about this.
Congratulations.
Thanks.
So your recent work is on read correction, right? Mm-hmm. How did you get to that? What was it, your first subject inside computational biology, or what was your path to read correction specifically?
My PhD thesis was focused on how to use the Bruijn graph to find new methods and new application of the Bruijn graph to handle some problem about sequencing data related to assembly. So to be more precise, I start to study the question on about how to align reads on the Bruijn graph, in a more general way, how to align sequence on a graph of sequence. Okay, so I focus on the Bruijn graph, and I build a tool that were able to align short reads from the next generation sequencing technology on the Bruijn graph, which is a graph used in genome assembly. And we got so many, it was really hard we were really excited about these methods, but it was really hard to get published because people were saying, okay, this is new, what are the application? And we were saying, we could think of application on assembly, on read correction, on compression, but people were saying, but that's in the future. So in fact, I started with this method and it was harder to publish. And right after, I started to do some work to show how this kind of method, so read alignment on graph, could be used for many applications. And here, I will be talking about read correction.
Yeah, so there is a lot to unpack here. You used a lot of terms that may be unfamiliar. So let's start with the read correction itself, right? It is what it sounds like. So you take sequencing reads and you correct the possible sequencing errors in them. But also, like, some people might think, oh, this sounds like a panacea. Like you could, so for example, in one of my previous podcasts, I talked to Cheng Xu about using unique molecular identifiers to correct sequencing errors in order to discover rare variants. And one might think, well, you don't need UMIs, you just correct the reads. It's as simple as that, right? But specifically, read correction is a problem that arises in the context of genome assembly. So maybe let's talk about the limitations of read correction, why it's not as widely applicable as one might think, but is specifically important in genome assembly.
Okay, so first of all, the main application of read correction is genome assembly. Because in order to perform a good correction of your reads, so we will be talking about next generation sequencing reads. So you typically got reads of hundreds of base pairs, so 100, 200, something like this, you will often got a very high coverage of your genomes. Maybe you will got 100 more nucleotides than your genome size. So you expect each region of your genome to be covered by like 100 reads. So you have a huge redundancy, okay? And in your NGS reads, you got approximately 1% of bases that are wrong, just because the sequencer usually make mistake. And using the redundancy of the information, you will try to detect that at this point, this read may be wrong. And you will try to change the nucleotide in order to match the other one. But of course, its application is limited because most of the time, many tools expect higher redundancy and try to handle those sequencing error by themselves. And there is another point is that most users are afraid of read correction because if the error correction creates k-mer, then it may impact or hurt the further analysis because we expect the noise of the sequencing data to be to be uniform. But if you correct, you may introduce a bias into your data, and many users are afraid of this kind of bias that you may introduce by correcting your reads.
Yeah, so if we think about some other uses of next generation sequencing, like maybe RNA-seq or ChIP-seq or something like that, first of all, you don't presumably have enough coverage to perform good read correction. And also, it may be just much cheaper to align it to the reference genome than to assemble the Bruijn graph, which we'll talk about in a minute.
Okay, you talked about RNA. That's a cool transition because actually I'm trying to see if we are able to apply the Bcool method into to correct transcriptomic data. The problem with transcriptomic data is you don't get this uniform coverage. So at some point you may lack good depth in order to infer what are the good sequences and what are the wrong sequences. So, yeah, here I would say it is really harder. It should be really harder to correct transcriptomic data.
Let's also formally define the problem of genome assembly. What is that and when is it needed? So probably a lot of people know about the Human Genome Project, which was highly publicized, but someone who works on humans may think, well, we already have the reference genome. Why do we need to assemble something else? Okay,
I will start by trying to define a genome assembly. So the idea of genome assembly is that you got, let's say you have a genome where you don't have a reference genome as, I don't know, a new species that you want to study because you are a biologist, for example. The first thing you are going to do is to sequence it. So you will use a sequencer, you will put some cells into the machine, and it will give you sequences, so A, T, C, G, things like this. But obviously, sequencers are not able to sequence a whole genome in one shot. So basically, you will get DNA sequences that are subsequences of your genome. And the trick is that you have no idea where these sequences came from in the genome. If you got the sequences, but you don't know if it's from the beginning, the middle, or the end of the genome. And the goal of an assembler is to get all those reads, all those subsequences of your genome, and try to reorder them in order to reconstitute the genome sequence. So it's like a puzzle, essentially. And after that, you have other problem like you have sequencing errors in your reads, so some bases may be wrong. And of course, sometimes you can't infer all the genome sequencing. So, for example, for the human genome, you don't have just one sequence. We have some parts that we call contigs or scaffold, and we are not really sure on how we should order them, on what is in what sequence are between the sequences.
Give us a very high-level overview of the modern algorithms for genome assembly. What do people use nowadays?
Okay, so nowadays most people try to perform assembly with what we call long reads. So this is reads that are that can count thousands of base pair. So basically, you will try to see which reads can overlap each other. And if a read share a very large overlaps with another, you will say that maybe after the read A, there is the read B. See what I mean? So using overlaps, you may you infer that the two reads are consecutive in the genomes. So you compute all overlaps into what we call an overlap graph, and you try to find what the genome sequence could be. And if when you are unsure, when you don't know if A should follow B or C, you should stop instead of doing a mistake.
Yeah, so that's called the overlap layout consensus algorithm.
Yeah.
Okay, so what are the alternatives to that?
For short read, for example, the most efficient way to assemble a genome is to use what you call the Bruijn graph. So here, it's because if you use short reads, you may got, for example, billions of reads. So if you have to compare each reads to all the other, it's really computationally expensive. So you don't want to compute all the overlaps between all the read pairs. Okay. So one trick that has been found is to use what we call the Bruijn graph. The Bruijn graph is also a sequence graph that is used to perform genome assembly. So the idea is that we are going to look at the words that are inside the reads of your sequencing data. So you are going to look at each word that we call k-mer, so word of length k. So you are going to look at all those words that appear in your dataset, and you are going to say if two words overlaps of k-1, if two words overlap almost completely, then they are connected in the Bruijn graph. So the Bruijn graph will be a graph where the nodes are k-mers, and the edges will be k-1 overlaps between k-mers. And the trick is that just by making this graph, you are going to the two reads that overlaps at least of k nucleotide will be successive into the Bruijn graph. So in fact, just by building this graph, you kind of compute the overlaps of at least k nucleotide between all the reads.
Right. So the Bruijn graph here plays two important roles. First of all, it lies under the genome assembly algorithm itself, but also your algorithm for read correction is also based on the Bruijn graph, as you already mentioned. Why do we need to correct sequencing errors? Why is it so important in genome assembly, and specifically in assembly based on the Bruijn graphs?
Okay, that's a good question, because it's especially important for the assembly with the Bruijn graph. So the trick is that the question is what is hard is genome assembly. In genome assembly, what is really hard to handle are repeated sequence across the genome. So if you have a sequence that appear at several positions in your genome, it will be really hard to assemble. Why is that so? It's because if you got a read, you don't know where it come from. So it can come from the first repetition, the second repetition, or the third repetition, etc. So in your graph, it will create a situation where you got a repetition that will got several sequence that can go before and several sequence that can go after. So you will have before one, before two, the repeats, after one, after two. So, and here you have no idea if your repeat is, for example, larger than your reads. You will have no idea how to connect the before to the after. And this is why some assembly will just have to stop. And this is why we are not able to perfectly assemble a genome. So repeats are repeated sequence across the genome are the reason that we can't perfectly assemble a genome. And in the Bruijn graph, the problem is that each repeat larger than k, so the size of the word we choose, so the k-mer size, each repeat that will be larger than k will be a problem in the Bruijn graph. Okay, so maybe a source of fragmentation. So you will want to use the largest possible k for the largest possible k-mer size in order to have as a low amount of repeat situation. So you want to use the largest possible k-mer size. And the problem is that you just can't choose a very, very high k-mer size because of the error rate. Let's say, you have let's say 1% of error. If you choose, k to 100, then you are very, very likely that all your k-mer in your reads contain a sequencing error. So the k-mer will not be a word that came from the genome, but will be a word that don't exist in the genome because it will contain, we expect that it will contain at least a sequencing errors. So if you use a too large k-mer size, your k-mer will basically be erroneous sequences.
Yeah, so when we construct the Bruijn graph, we sort of rely on the fact that the overlapped parts of different reads will collapse into a single node or a single edge in the Bruijn graph. And if you have at least one error in the k-mer, it's already a different k-mer. So it's a completely different node in the Bruijn graph, and this collapsing doesn't happen. And this is what is important for the assembly algorithm.
Yeah, and if you have, let's say, 1% error, so you will have to choose a k-mer size adapt to this error rate in order to get enough genomic k-mer, so k-mer that don't contain an error, in order to be able to build the Bruijn graph that contain hopefully all the genomic k-mers and contain a low amount of erroneous k-mer. And let's say if you didn't get any sequencing errors, then you could, or a very low amount of sequencing errors, you could use a larger k-mer size, and then you will get less repeat pattern, and that you will be able to build a more contiguous assembly. Sorry, I just forgot to say that I spoke about the Bruijn graph, but even so at the beginning we didn't use the Bruijn graph to assemble genome. We were computing overlaps between all the reads and compute an assembly like this. And even in this situation, even using the overlaps layout consensus paradigm, it is very useful to correct the error that are in the reads, because if there is a 1% error rate, then you have to compute overlaps with an edit distance between the possible overlaps. But if your reads contain a very low amount of sequencing errors, then you can ask for extremely precise overlaps, so you can go faster by finding relevant overlaps. So in fact, at the beginning of the short read assembly, we were correcting the reads in order to be able to focus on very precise overlaps between the read and to perform this crucial step faster.
Yeah, so the need in error correction was recognized very early on, and the early papers on the genome assembly already contained some simple algorithms to perform this kind of error correction. So what were the early methods for error correction? So
in fact, there were three essential categories to correct reads. There were the use of a suffix array. So you would construct like a suffix tree of your reads, and you would try to find if your reads would match to the suffix of many other reads, and if you were close to a sequence that were highly present in the set, you would try to correct them. There were also techniques based on multiple alignments. So basically, you will have to align many reads together and try to find if they all agree on a sequence, and if there is one read that say A while the others say T, then maybe that's a sequencing error, and we will try to substitute the A for a T. But the most used methods nowadays is based on k-mer spectrum techniques. And even now, when you read a review, most techniques and the more efficient technique are based on the k-mer spectrum ID.
Yeah, and k-mer spectrum is also pretty easy to explain compared to the other two, right? So why don't we just try to explain it to give some understanding of what was popular before, or even right now?
Yeah, that's in fact, k-mer spectrum is a very, very simple idea. The idea is that when you are going to look at the k-mer of your read file, and since your read file is highly redundant, you expect most genomic k-mer to be present. Let's say you have a coverage of 100. You expect most genomic k-mer to be present, like, I don't know, 60 times, 70 times, 80 times in your sequencing data. So you are going to count all the words that appear in your read set, and then you are going to see that some k-mer will be highly abundant in your, and those are very likely to be genomic k-mers. And on the other hand, the erroneous k-mers will be present like 1 time or 2 times if you are not lucky, because they are just noise. You see what I mean? You take a genomic k-mer and you modify it, but there is a priori no reason that it match another genomic k-mers. So just by looking at the k-mer abundancy, you may be able to distinguish genomic k-mers because they occur a lot in your read set, and erroneous k-mer because they will occur just 1 or 2 times. So the idea is that you are going to separate your k-mer in 2 categories. The first that will be called solid, because they appear a lot in the read set, and the other one that will be called weak, because they occur just few times. So you will have some trusted k-mers and some k-mers that will not be trusted. Okay, so once you have inferred this set of trusted or solid k-mers, you are going to look at your reads, and you are going to see that some k-mer of your read will be trusted, and some will not be trusted. Because if you've got, when you will have a sequencing error, all the k-mers that contain the sequencing error will be, most of the time, weak. Then you will be able to locate the sequencing error because it will be covered by weak k-mer, and then you will try to change the suspected nucleotide in order to cover your read by solid k-mers.
And so regarding this technique of k-mer spectrum read correction, in your paper you point out 4 possible issues with it, right? So weak genomic k-mers, solid erroneous k-mers, errors covered by solid k-mers, and nearby errors. Let's maybe walk through them and explain in a couple of words what those issues are.
Okay, so in fact, you may make at the beginning you may make 2 kinds of mistakes. For example, the first mistake may be to if there is an erroneous k-mer that is seen many times, because it originates from a highly repeated sequence in the genomes. So an erroneous k-mer may be trusted. Okay, so I call this case the solid erroneous k-mer. Okay, and the problem is that if you look at your read, you will see that you will not see any problems, because you are going to look at the erroneous k-mer and you say, okay, no, this k-mer is trusted because I have seen it many times in my read set. So you may not correct this kind of sequencing errors. So this is one case. The opposite case is the weak genomic k-mer. So let's say your coverage is not that high, and you have a k-mer that is genomic, that appear in the genome, but that occur just a few amount of time in your read set. So here, this is the opposite. You will look at a reads that don't contain sequencing errors, but you will see that some k-mer will be untrusted, and you may try to correct some sequences just because they are not covered enough by the other reads of your dataset. So those are the two, let's say, easy errors. So basically, if you got a very, very abundant dataset, you should not have this kind of problem, because the higher the coverage, the easier it should be to distinguish genomics from erroneous k-mers. But there is another problem that occur around a repeated sequence. Let's say you got ATCG, and in the other part of your genome you get ATCC, okay? And if you got sequencing errors on the G, and the G became a C. So in fact you got a sequencing error, but the ATCC is also a genomic k-mer, because it appears in another part of your genome. So you will trust this ATCC, despite the fact that it contains sequencing errors. You see what I mean?
Isn't the same as solid erroneous k-mers, or what's the difference?
No, because here, so you have ATCGs that become ATCC, but yeah. ATCC is a genomic k-mers because it appears in the genome. So ATCG and ATCC looks alike because there is a repeat that is ATC, and by chance the sequencing error change a genomic sequence in another genomic sequence. So you don't detect the errors because you are just looking at the word and not at the context.
Okay, so the solid erroneous k-mers arise not because these alternative k-mers are also genomic, right? They arise for some other reason. Because
you have seen this specific k-mer many times in your datasets. Because you
just by coincidence.
Yeah, by chance.
Okay.
And here the problem is that ATCC for example will be seen many times in genome because it is a word of the genome. And this is the problem here is that ATC is a repeated sequence in the genome. So the more repeated your genome will be, the higher chance you will have to get this kind of patterns. For example, we took an example of lighter, so a very good error corrector. If you run it with simulated data on C. elegans genomes, and at the end, after the correction, you will only get 3% of reads that still contain an error. So almost a perfect correction. But if you run the same algorithm on the human genome, so a more complex and repeat-rich genome, the number of reads that contain at least an error after correction is 40%. And in fact, the more complex, I mean repeat-rich, your genome will be, the harder it will be to correct this kind of sequence.
Right, and so you attribute that high error rate to the repeats.
Yeah, because the problem is that it is hard to correct around the repeated sequence of the genomes. When you are close to a repeated sequence, it will be hard to correct the error because the k-mer spectrum just look at the word of the genomes and don't look at the context.
Right, that makes sense. Okay, so we covered the three of the issues, so weak genomic k-mers, the solid erroneous k-mers, errors covered by solid k-mers because of the repeats, and the fourth one is nearby errors. What is that about?
This is another problem of a k-mer spectrum, is that they expect most sequencing to precisely locate and isolate, for example, let's say one sequencing errors. And since so they are searching for, so one sequencing error will create k-1 contiguous erroneous k-mers, if on the perfect cases. But if you got several sequencing errors, it will be hard. You will get a large region of weak k-mer, and it will be hard to know how many errors there is inside these regions, and how to correct them efficiently, because you know that there is many sequencing errors in the zone, but you can't tell how many, and you are not really sure on how to correct them, because you don't trust any basis that is covered by sequencing by weak k-mers. But between the sequencing errors, you may have accurate base, but you have no way to know for sure that they are accurate.
Right. So normally, when you correct the reads using the k-mer spectrum, you have this k-mer that occurs very few times in the spectrum, and you look for the k-mers, other k-mers that are solid and that are maximally similar to this one, right? So you're looking for the lowest number of changes that you need to make to this k-mer to get some other k-mer, right? And so you're saying that when this number of needed corrections is high, then that presents a problem.
The problem is combinatorial. You have to, theoretically, you should try to every possible substitution in the region, and if the region is large, then you should check many k-mer solidity, and in fact it may be intractable if the region is large. So most k-mer rely on greedy heuristics, but I mean those are greedy heuristics.
I'm wondering, it's very similar to a simple mapping problem, right? So you could use the same techniques from the mapping problem, like seed and extend.
Yeah. In fact, that's quite similar to mapping. We are searching for the minimal distance to make it look like something we know. So that's look like an alignment problem. And in fact, the corrector of Engli called BFC try to explicitly correct a read as a whole by finding the minimum edit distance to match to only things that are considered correct.
I wanted to ask how you arrived at the De Bruijn graph correction as a solution to this problem, but from what you told me before, it more sounds like you already had this method and you were looking for the applications, right? Yeah,
I've made a method able to align reads on the Bruijn graph, and my point was that the Bruijn graph, it's quite easy to construct a good De Bruijn graph containing most genomic k-mers, and while containing very few, a very low amount of erroneous k-mers. So in fact, the Bruijn graph can be seen as a reference that is quite easy to construct. Because, if you want to infer a set of trusted k-mer, you just rely on the abundance. You see what I mean? You just say, okay, this k-mer is low, so I don't take it. This k-mer is seen many times, so I take it. When you build the Bruijn graph, you can include many k-mers, even let's say k-mers with medium abundance, and you can look at the graph and detect k-mers that are very likely to be sequencing error, despite, for example, their high abundance. So in fact, it is easier to build a good, to infer a set of genomic k-mer using the Bruijn graph than just the k-mer spectrum, because you have a topological aspect on the Bruijn graph that you don't have on the k-mer spectrum.
Maybe let's start with how you build the De Bruijn graph, right? So you just, for every read, you extract all the strings of the length k+1, and each such string gives you essentially two vertices. That's the first k-mer and the last k-mer, and the edge between them. So you just add those to the graph, and once you add everything, and vertices, they collapse if the same k-mer occurs multiple times as the single vertex. And there you have your De Bruijn graph, right? Then you do some post-processing on the graph. You do some kind of cleanup, right? Can you talk about that?
Yeah, I can talk about that. So the idea is that the De Bruijn graph is a graph that's trying to order the world of the k-world, the k-mer of the genome. And the very good property of the De Bruijn graph is that just by looking at the De Bruijn graph topologically, you are going to see many things that are very likely to be sequencing errors. For example, if there is a sequencing error at the end of a read, it will create a small dead end in the graph. That's a path that don't lead anywhere. And you don't expect to see a short path that don't go anywhere, because the Bruijn graph should stop at the end of a chromosome. So you don't expect to see a short tip like this. So you can just remove this kind of patterns, and just by doing that, you are able to remove most sequencing errors, for example.
Yeah, that's a very neat idea. And the idea there is that the sequencing error, when you look at the De Bruijn graph, it could result in either an alternative path. There's sort of like a loop, or I don't know, how do you call it, a bubble?
A bubble or a chip, yeah.
Yeah, or it could result in this dead end.
Yeah, because it will not connect to the other genomic sequence, because it is another sequence that only comes from the genomes.
Right. And the difference between those two is that if the error is somewhere in the middle of the read, which is separated from the ends at least by K nucleotides, then you can transition into the error via correct k-mers, and you can transition out of the error via correct k-mers, and so it becomes sort of integrated in this de Bruijn graph. But if it's close enough to the end of the read, then you can get into it, but you can't get out of it by a correct k-mer, right? And this is where this idea of using the high value of k comes into play.
Yeah.
Because the higher your k, the higher the probability that the sequencing error will be at most k nucleotides to the end of the read.
Exactly. With a higher k-mer size, you will get most of your sequencing errors that will become a tip. But so the tip is really the easy errors to remove. But if the sequencing error is in the middle of the read, it will create a bubble. And here you will see a bubble in your graph with two paths, and you know that one is incorrect and the other one is correct. So the question is, how do you choose? And here we rely on what I call unitig, or should I say path abundance. So we are, if in the k-mer spectrum, just say if this k-mer, if a k-mer is seen more than 5 times, then I keep it. If not, I just reject it. We are doing something a bit smarter. Let's say we are going, so the two paths, we are going to compute the average abundance from each path. So we compute the average abundance of the erroneous path and the genomic path, and we expect the average abundance of the genomic path to be very high compared to the erroneous path. Because, of course, those who came from the genome should be highly abundant.
Right. So when you're looking at the k-mer spectrum, you don't have any context. You see only this k-mer, but you don't see the neighboring k-mers.
Yeah, you don't have the context. Exactly.
But in the de Bruijn graph, you could average is the same number. So the abundance you're talking about is just the number that this k-mer occurs in your sequencing data, but you could average it across several neighboring k-mers.
Yep, exactly. And this has two advantages. The first advantage is that you may save a genomic k-mer that has a low abundance, but which got neighbors that have a high abundance. And you could also remove an erroneous k-mer that have medium abundance, but with its neighbors that should have a low abundance. Just because we average the abundance on the simple path.
Yeah, so this estimate becomes more robust, more reliable.
Exactly. So it's most robust because we use more context than just a raw k-mer abundance filter.
So do you have some kind of threshold for the average abundance, or do you always remove all but one paths?
Okay, to be very precise with first, so we build the de Bruijn graph. We may remove, for example, k-mers that are unique because we really don't believe that we should keep them. Then we perform tip removal, and then we compute the abundance of each simple path of the graph. And we apply this threshold on this kind, on those simple paths. So on the average abundance. Yeah, that's a threshold, and you could infer the threshold just by looking at you could plot the distribution, see unitics that have so plot the abundance distribution and see and try to infer which threshold could be a good separation just by looking at the spectrum.
And so at this point, you have a choice. You could either try to use this graph to correct your reads, so align the reads on the graph, or you could say, well, I already have a de Bruijn graph and I already cleaned it up. And so I have this clean de Bruijn graph. So why don't I just move on to the assembly?
That's a very nice question. Most people say, yeah, you have a de Bruijn graph, so basically you are performing assembly. But in assembly, sometimes you may... It depends on the way you want to assemble. For example, some people want to ignore the heterozygous information and crush the bubble that are generated, let's say, by variants in your genome, if you are diploid, for example. And some say, okay, we want to keep it. So just building a de Bruijn graph for me is not performing assembly. It's the first step. And in fact, we believe that mapping the reads on the de Bruijn graph could produce a good assembly, but that's another project of my group.
Yeah, and also the reads themselves contain extra information that is lost when converting to de Bruijn graph. So if you correct the reads, right, then while assembling, you could check if, for example, all the corrected reads actually occur in your assembly, because you could arrive at the assembly from a de Bruijn graph which is consistent with the de Bruijn graph, but not consistent with the reads you have, because reads inherently have more information because they're just longer than your k-mer.
Yep, exactly.
How does the process of aligning the reads on the de Bruijn graph, how does that look like?
Okay, so here there is no real novelty. It's kind of a seed and extend strategy. So basically what we are going to do is to index some anchor, so sequence that appear in the de Bruijn graph. So the important point is that those anchor can be smaller than k. That's the important point. And so let's say 21, 31 as a parameter. So by default, I think it's 21. So let's say 21. We are indexing the 21 mares that appear in the de Bruijn graph. And we remember at which position, so where in the de Bruijn graph they appear. And to align reads on the de Bruijn graph, we are searching for anchor in the read. If we find anchor, we know that there is the beginning of a possible alignment, and we will try to extend the alignment and to see if it results in a good mapping with let's say a naming distance of 1, 2, etc. And we will try to align the read on different paths, considering its anchor, and keep the best alignment.
Does the alignment have to be on a simple path, or could it span across the branching points?
Thanks. That's really a good question. At the beginning, some people would say that we could just align the reads on the simple path of the de Bruijn graph. But in fact, this is not enough, especially for complex genomes where the de Bruijn graph is really fragmented and where most simple paths are smaller than a read. So in fact, we can start at let's say at the end of one simple path, and then we'll try to extend the alignments and by choosing the neighbors of a node. So we will try to extend on the node that continue with A, and we will also try to extend with the nodes that start with T, for example. And we'll keep once again the best mapping.
Previously, we talked about the 4 issues with the k-mer spectrum method, and I think we already covered some of these, but I think it's just useful to go through them in order and to say how your method fares better on those issues than the k-mer spectrum. So first of all, weak genomic k-mers.
So in fact, the 2 first problems, the weak genomic k-mers and the solid erroneous k-mers, so those are just the problem of inferring k-mers that are genomics and infer which k-mer are not genomic. And in fact, just because we build a de Bruijn graph and we use a topological heuristic to remove sequencing error and to keep genomic k-mers, we have shown that we were able, using the cleaning of the de Bruijn graph, to remove more sequencing error and to keep more genomic k-mers than just than a k-mer spectrum approach. So in fact, we have less weak genomic k-mers because we keep more low abundance dynamic k-mer, and we have less solid erroneous k-mer because we remove more k-mer that may be of medium abundance. So in fact, those two problems are not solved, but less encountered because we build a better state of k-mer using topological heuristics using the Bruijn graph.
Yeah, and also the averaging helps, right, to determine the abundance.
Yeah, and this is because we use the Bruijn graph that you can average using the neighbors.
Yeah.
This is because we have a topological argument to say that.
Right. And so what about the errors that are covered by solid k-mers because of the repeats?
Okay, so here this is because of the mapping. So the main problem of k-mer spectrum technique is that they have a very good practical value that is 31. Because if you use a k-mer spectrum, you are not able to use a large k-mer size. Why? It's because if you use a very large k-mer size, most of your k-mers will contain sequencing errors. So you will have less errorless k-mer, and it will be harder to locate error because you have less errorless k-mer to rely on. And the larger the k-mer size you will choose, the more very hard cases you will find, like multiple errors in k-mers or reads that don't have any errorless k-mers. So in fact, if you try to use a k-mer spectrum with a high k value like 63, it will not work at all because the very good practical value of k equal to 31 fits the error rate of the NGS dataset. If you go higher, you will not have enough errorless k-mers, and if you go lower, you will have more repeated, more problem related to repeats. So this is a huge problem of the k-mer spectrum. For me, it's really a limitation of the k-mer spectrum correction. But if you use the Bruijn graph, then if you have a very covered dataset, then you may be able to construct a Bruijn graph with a very high k-mer size, like say 60, 70, 80, and you can even go higher if you have very good coverage. But this is because our alignments, in order to be able to correct a read, we should align it on the graph. And in order for the alignment to work, we just need to find an anchor that don't have sequencing errors. So, and here the anchor is not related to the k-mer size. So let's say your k is 63, and the anchor you will use is maybe 21. So if you correct by aligning on the Bruijn graph, you need, let's say, a 21 mer that don't have any sequencing errors. When you want to correct with a k-mer spectrum technique, then you need multiple k-mers that don't contain errors. So in fact, using our Bruijn graph, we are able to use very high k-mer size, and then to be less sensible to a repeated region, and then to perform a better correction.
Right, and so the last point is nearby errors. How does the Bruijn correction deal with those?
Yeah, here, this is the best example of why the alignment is easier. Because we have the context, if there is like 3 errors that are in a row in our alignment, it don't change anything, because we will say, okay, we have this mapping that got 3 errors, but it don't change anything. We will just choose the best, the alignment that got the lowest amount of errors. But since we have the context, we could say, okay, even if there is 3 error here, it's still the best mapping, so we should keep it. So here the problem is non-existence, since we align the 2 sequences together, since we have a reference and the read.
Yeah. So your query is just larger. You have the full read as opposed to a single k-mer.
Exactly. And I really want to insist on the fact that the main, because in the paper we show that we are able to perform almost an order of magnitude better correction, but for me, the main reason is that we are able to use a higher k-mer size, because once again, to correct a read, we just need to be able to align it. So we need an encore that is errorless, while the k-mer spectrum need a k-mer that is errorless. So k-mer spectrum can't use a high k-mer size, so will be sensible to repeats that are, let's say, medium sized. And if you use our method, we are able to use larger k-mer size and then to be less sensible to the repeat problem. And this is why we have very good results, especially on a genome like the human, where you got many repeated sequence, because we are able to use a larger k-mer size, so we are less sensible to the repeats. I think this is the main point.
So we mostly talked about correcting the base calling errors where you just switch one base for another. Do you try to deal at all with insertions or deletions, which are not that common in the short read NGS data, but perhaps more common in the long read data?
Okay, so there is two things here. First, it is interesting to align. It should be interesting to align on deletion and considering insertion and deletion, because you have the Pacific Bioscience reads that provide short, accurate reads that contain insertion and deletion, and we may want to correct them using this kind of technique. But we have no technical limitations that the alignment steps that I made, that is called a big rate, do not handle yet insertion or deletion. So we are just computing Hamming distance. So we will never consider insertion and deletion. So for now it is impossible, and because at the beginning we were really focused on the correction of Illumina reads that mostly contains substitution. But it definitely should be interesting to correct, let's say, CCS sequence from PacBio. But you were talking about the long reads, and here I think it's just another application, because the long reads contain a very high amount of error, so I don't think our alignment should be satisfying. So we should develop a new aligner that are able to align long, noisy reads on graph, so maybe the Bruijn graph, maybe on another sequence graph. But yeah, that's basically perspective we should explore.
And also in the paper, you talk about the algorithmic approach to the selection of the k-mer size. Could you talk about that?
Okay, so here we want to use the highest possible k-mer size in order to have a low amount of repeat in the Bruijn graph, and in order to be able to get a very the best possible correction. So our goal is to use the highest possible k-mer size. But if we use a too large k-mer size, we may have hole in our graph, and we don't want that. So it's really a trade-off, because if we put k too low, okay, we will have all the genomic k-mers, but we may include some repeats, and if the k is very high, we will have less repeat, but we may miss some k-mers. So the idea is, should the user select a k-mer size, or should the software choose himself? Because the size of k selected is really critical for the performance of the tool. So the idea here is that we are going to plot several k-mer spectrum and select the one where we expect to be able to perform a good separation, and we will select the ones with the higher k-mer size. Because at some point, if k is too high, then you will not be able to distinguish the two peaks. Because in a k-mer spectrum, we expect one very large peak of erroneous k-mer, then kind of a calm belt where you have almost no k-mer with a medium abundance, and then you expect a second peak with all the highly abundant genomic k-mers.
Right. So the two peaks correspond. The first peak corresponds to roughly to the error rate,
yep.
And the second peak corresponds to the coverage.
And if you use a larger k, the two peaks will be closer. And at some point, they will be almost merged. And here, you will not be able to say if a k-mer is an error or a genomic k-mers. And if you use a very low, basically, if you use a low k-mer size, you will be able to perform a very good distinction, and the higher k, the closer the two peaks will be. So we will plot several k-mer spectrum and try to select the one where we are confident that we will be able to separate the two. And still, we want to use the largest k-mer size.
Yeah, and that procedure does not only give you the k-mer size, but also gives you the threshold that you can use to separate those and to filter out the rare k-mers.
Yeah, and it's highly inspired by k-mer Genie. That is an analysis of a k-mer spectrum in order to select the good k-mer size to use to perform the Bruijn graph assembly. And of course, so basically we made a k-mer Genie that is that performs a somewhat simpler analysis.
Well, this sounds very cool, like almost revolutionary, right? Are there any caveats? Because it almost sounds too good to be true. Are there any downsides to this algorithm?
Okay, there is one downside compared to the state of the art, is that k-mer spectrum techniques are extremely efficient, like really, really, really efficient. I have an example where Lighter is able to correct 100x human datasets using 10 gigabytes and 5 hours. So it's lightning fast, because they perform extremely simple operation, in fact. So they just have to index words in a set, like a Bloom filter or something like this, and then you just perform query on the k-mer of the read. They can do it in parallel, etc. So they are extremely efficient in both the time and memory efficient, while Bcool have to construct the Bruijn graph, clean it, and then align the read and then. So I would say that the caveats of Bcool right now is that it's not that efficient. We are still able to correct the human genomes, but it uses several more memory than any other tools.
So how long does it take?
So we have an experiment on the human genome where it took for 100x, it took like 1 day to correct a 100x dataset from the human. So it's still
okay. That's not too bad, right? 1 day compared to 5 hours.
No. That's not too bad. I mean, we are quite satisfied with it, but it is not lightning fast like the state of the art.
Especially since then you still have to perform the assembly itself, which is presumably not exactly fast, right?
Yep. And another, let's say a personal criticism is that most assemblers are made to manage, let's say, 1% on 1 per 1,000 error rate. So in fact, even if you correct very well your read, it don't make that much of a difference because a k-mer spectrum, even if it divides the number of sequencing error by 10, even if Bcool divide the number of error by 100, it don't change that much, because most tools are in fact able to handle some sequencing errors. Mm-hmm. So just performing a really, really better error correction don't help that much, because most tools are in fact made to handle those errors.
Yeah, it's like the law of the diminishing returns.
Yeah, exactly. But I'm quite confident in the fact that for larger genomes, like, I don't know, I worked on the Pine genome that is 20 gigabytes long. Or, I don't know, we may have heard of Paris japonica, that is a flower, that is the largest sequenced genomes, that is 150 gigabytes long. So a huge genomes. So we found a chromosome in Paris japonica that is in fact larger than the human one, the human genome. So I'm confident that on this very large genome we will see the interest of a better error correction, because I expect that k-mer spectrum will perform quite bad on those genomes.
Oh, very cool, Antoine. Very impressive work. And we'll see if it gets incorporated into all the major assemblers. That would be cool.
Yeah, that would be cool. Yeah.
All right. Well, thank you so much for talking to me today.
Thank you for listening to me.